the research that I've been doing. So I work out at Kennedy Space Center in the Space Life Science Laboratory. I'm kind of with the University of Florida and with the NASA Postdoctoral organization out at KSC. So we do a lot of collaborations together, and today I'm gonna kind of share with you some of the research I've been working on the last 2 years, which is trying to figure out a way to identify the plant microbiome for ISS application. All right, so I'm going to share with you a few things. One is basically how plants respond in the spaceflight environment, what kind of studies we have done to try and further our understanding about plant growth in different regoliths, like lunar regolith, Martian regolith, some of the understanding of how the gene expression of plants change. This is things that I was working on during my doctoral studies. We looked a lot about the the molecular aspect of plant development in the ISS and in microgravity simulated environments. And so we really tried to understand how the plants are responding, what genes are turned on, what genes are turned off, and then also use some of that skill set that I developed during my doctoral studies and push them toward plant microbiomes, trying to figure out what the functional activity of microbes are, and who's there and what they're doing for the plants. All right, so, and I'm sure all of you are very well aware that we are going on this Artemis mission. Artemis mission to the moon is going to be sometime, in the 2024 area, and the journey to the moon is going to help us prepare for our journey to Mars, which is going to be somewhere in 2030s. There's so much that we need to know, so much that we need to learn, and we're going to use the moon as a step stool towards our eventual goal of colonizing Mars. So, as I said, in 2030, NASA plans to have a manned mission to Mars. So we are going, but the journey to Mars is going to be a really long one. And once we get there, we need to create a sustainable environment for us to live in. So how do we do that on a rocky, barren planet? Then, the answer is we need to bring the most versatile organism on Earth with us, and that's plants. So I'm sure most of you, from the introduction, most of you are aware that plants are really important. Plants will provide us with oxygen, with carbohydrates, food, water, and to recycle waste. And so this is going to help us create a sustainable environment when we get to Mars. So plants are really important. And so I'm sure you've seen the movie The Martian, and here are some of my favorite quotes from that movie. And I'm sure you're going to, I could be a little biased because he's a botanist, and I like plants and I study plants. So these are some of my favorite quotes. If you haven't watched the movie, it's basically an astronaut getting stranded on Mars, and he needs to figure out how to survive. So one of the things he did in the movie to solve his food crisis, he grew potatoes on Martian soil. Being able to grow plants on resources available on a planet would be wonderful, would be excellent. However, there's still a lot of experiments that we, as in the scientific community, have to conduct to really understand how well plants can grow on Mars, and what is feasible, what is not. From my personal experience, we have played around with some of these Martian simulants. these things, these stimulants that we use were grown from, were collected from the volcanic soil in Hawaii. And it is known to be really similar to the Martian soil composition. So in a few slides, I will show you some examples of what we have done in the lab. So these experiments that we've done basically found that plants can grow in the Martian stimulants, basically soil from the volcanic soil from Hawaii. So there's still a lot of things that we have to figure out, though. This is not exactly a one-to-one comparison, because volcanic soil from Hawaii is, though it's kind of similar in composition, it's not exactly the same. So there's still a lot of other considerations that we need to take into, that needs to come into play here. But in terms of growing plants on Mars, another thing that we want to consider is the weather. The weather in the environment in Mars is extremely cold, and plants can't survive just growing out in the open. So what we're thinking, what NASA's thinking, is controlled environment systems. So a lot of greenhouses that will probably be used to try and grow plants when we get to Mars. So here, let me share with you some of the things that we envision when we get to Mars. Well, how are we going to set up systems to grow plants? These are some of the examples that NASA has envisioned when we go to the moon or when we go to Mars. Everything is in a controlled environment, greenhouses. A lot of hydroponics is probably what we're going to use. But there's still a lot of things that us at KSC are trying to figure out, you know, how well, what nutrients, what plants, you know, what the plant microbiome in the hydroponic system is going to be like. A lot of the times what we use is this arsenite. It's like a clay substance that we use to grow our plants. This is what most of our current experiments are done in. But the idea is to move into more hydroponic system, and we're still trying to figure out the best hardware, the best tools. So there is a watering is an issue for us, because when you don't have gravity, the water tends to accumulate the base of the soil, and that leads to plants experiencing this flooding stress. And so this is something that we want to avoid, and we're trying to figure out the best hardware to use to help. So there's current experiments trying to understand how passive watering is going to work, how active watering is going to work. And so there's a lot of development in this area. So one of the things that I'm going to share with you is some of the experiments that we've done here on Earth to help us get to to understand how plants can adapt to this novel environment of spaceflight. We do a lot of planetary analogs, and this was during my time as a doctoral student at the University of Florida. The two planetary analogs that we have done experiments in is one in the Arctic and one in Antarctica. So in the Arctic, Devon Island is one of the places that we actually had a remote greenhouse, remote control greenhouse. And I will share with you a story about that greenhouse. It's a sad story. I'll share that with you in a bit. So in the Arctic, this is the place they call the Halton crater on Devon Island, and it is a remote place. It really looks like Mars over there. They have a Mars analog area, and you can see the similarities between Halton crater and on Mars. And here I showed you some pictures of some of the simulant that we have collected from that area and tried growing plants in. Over there, we actually have a remote sensing greenhouse that was built, I want to say about 6 years ago. A group of the group from the University of Florida with Dr. Rob Frohl, Dr. Annalisa Paul, Matt Bamsey, and Tom Graham. They went up there, built a greenhouse, and we were trying to use remote sensing to grow plants. So the idea is, if you do go to Mars, you can't bring everything with you. So the idea is to deploy such a greenhouse on Mars so that the plants can grow without any human intervention or any human handling until the astronauts do get there. So the crops will already be growing, and the astronauts will have food available when they do go onto the planet. So the greenhouse was built. There was 3 years of actual active study in that greenhouse, and then the very last year, a polar bear came and tore the entire greenhouse down. It destroyed all the equipment that was there, and that was the end of that project. So it was kind of sad, but such is the life of science, I guess. You can't control everything. So the other cool thing about Martian simulant that we have tested as well is one that I mentioned from Hawaii, and the other one is from the Mojave Martian simulant. So we've tested both systems, try to grow plants in them, and plants do grow, but they don't grow as well as the soil, the normal soil that we use as controls. I think someone in the audience had quite a bit of experience growing plants on Martian simulant. So maybe you have a different outcome. Ours didn't grow very well, so that's there's still a lot of things that we're trying to.
Natasha, I've got a question for you. Could you tell us a little bit about how the plants didn't perform so well? Like in what ways they underperformed?
Yeah. So, for example, let's see. Over here, we have this particular one is plants grown on your regular soil mix that we use in the lab, and you can see some of the examples that we have here. They are really much smaller. These are all grown at the same time, at this tentatively supposed to be at the same developmental stage, but you can see they're so much smaller. There's a delayed growth, and they don't grow. So yeah, that's some of the examples that we have when we're trying to play with the Martian simulant. But there's still a lot of active research going on at Kennedy Space Center, where they're using different components. They're adding more nutrients. I know also that they are testing, well, at least in the University of Florida, they were testing some genetic engineered plants to see if they can deal with magnesium sulfate. Yeah, so there are a lot of active research going on in this area.
So I thought hydroponics is growing plants without soil. So if the idea is to not use soil and use hydroponics, why would there even be this work with simulated Martian soils?
Well, so the thing is, we want to, test out as many different possibilities so that, if one doesn't work, if something goes wrong, you'll be able to know what your backups are. So for Martian soil, they're testing whether they can use the resources there to grow plants actively without, hydroponics. But at the same time, we are also developing hydroponic system. And, even like a nutrient film system, so that we can do different, large scale greenhouse, and then eventually, if we figured out how best to grow plants on the Martian soil, then we can start growing them as well. You just want to make sure that you have a lot of different options and you want to test them all. So yeah. So the other analog that we have is also on Horton crater, and this is the Lunar Analog site. So in that one crater, they have one part that is Martian and one part that is lunar, which I thought was really fascinating. And so again, we have been trying to grow Arabidopsis on the lunar simulant as well. And again, they to some degree they do grow, as you can see, but not the same. And there is still a lot of things we're trying to figure out, like what nutrients need to be added to it. Do we genetically engineer these plants so that they can adapt to that kind of chemistry from the lunar simulants? A lot of things we still don't know. Everything that I'm showing is kind of, we're testing and we are playing around with different aspects and we're trying to figure things out. And so another one is in Antarctica, and this is called the Eden Iss. And the Eden Iss is actually ESA developed. I think, yeah, the PIs in this project are down in Bremen, Germany. And they've converted a shipping container into a greenhouse. So we're going to remote places that is hard to grow plants, and we're trying to figure out how best to do it. How we have the greenhouse in the Arctic, we used Martian simulants, we use hydroponic systems over there as well, we use lunar simulants. And now in the Antarctica, we're doing the same thing. We're trying to figure out, mostly in hydroponic system, how best to grow the plants. How much space do you need? What kind of volume? What kind of yields you're getting? How is it going to sustain? How many people is it going to sustain? All these kind of questions are trying to be answered right now in the Eden Iss module. So then there is also different kind of angle where we're trying to figure out how gravity affects spaceflight. How gravity affects plant growth. So a spaceflight analog is this parabolic flight. And we do grow quite a bit of plants, actually quite a bit of experiments. In the University of Florida, Rob and Annalisa have had close to 20 years of experience with the parabolic flight. So basically, a parabolic flight, you have your 2Gs where you're climbing up for 20 seconds, and then you start free falling, and you experience 0Gs for 20 seconds. And you do this 40 parabolas. And so here, I don't know if you can see, but here's a video of us doing a spaceflight experiment. Well, not spaceflight, sorry, parabolic flight experiment. And what we have is a hardware over here that we grow our plants in, and we're trying to see how they respond to to 0Gs and to 2Gs. So you can see in some of the experiments that we have that plants do have a very rapid responses. They can tell when they're experiencing 0G and when they're experiencing 2Gs. You can see thermal changes in the leaf area. You can see gene expression changes in the roots and in the leaves. So they respond really quickly. They're very sessile organisms, and so they have evolved methods to react really quickly to the environment, which is really interesting for us. Yeah, so we also do have quite a bit of fun on the Parabolic Flight, which is really, really awesome. Yeah. All right. So yeah, it's pretty fun to test out how plants respond in all sorts of different environments that relate to spaceflight as well. So I showed you a few of the things that that we tested out during my doctoral studies. So we went up to the Arctic, tested out different stimulants. We had an expedition to the Antarctica, and we did a lot of parabolic flights as well. And all of these led us to figure out, the molecular response of plants, how the genes change in these different environments, how the plants grow. Are they healthy? Are they growing well? What kind of gene pools are turned on? That can also help you figure out what stress response they are dealing with in these novel environments. And then you could also figure out what's lacking, and you could compensate what's lacking with genetic engineering. So here is basically how one of our spaceflight experiments is conducted. We grow our plants. So these are Arabidopsis plants on Petri dishes. This square Petri dish, they have a gel matrix called phytogel, and phytogel has all the nutrients and water content that the plants need to be healthy and to develop and grow. So what we do is we prepare these plates. We have seeds that we plant on these plates. And as you can see here, the seeds are really tiny for the Arabidopsis plant. What we do is then we wrap all these plates in a cloth, in a light, in a dark duvetine cloth that basically prevents light from entering into the plate. And so this is important because light is the cue for the seeds to start germinating. And we don't want the seeds to germinate before they get onto station. So these plates are all wrapped up, and what happens next is they get launched. For this particular set of experiments we did, that was in 2009, 2010. So they were launched by on the STS, I think 129 shuttle. And so then they are put in the hardware on station. Then the astronauts unwrap the cloth, put them in the hardware. Light is, the plants are exposed to light, and they start germinating, and they grow in microgravity. And so astronauts help us with our experiments. They take the plants, they harvest them into tubes called Kennedy fixation tubes. They're called KFTs. And so basically, once the plants are grown for a set amount of time, and we track the development of that plant, we take photographs, we see, we basically make sure that that plate has a lot of germination. The astronauts basically will then harvest the plants and then put them into the KFT with RNA later. So it fixes the plants. It makes the RNA stable so that we can then bring it back home and extract the DNA and RNA and start investigating, the gene expressions, how that changes in flight. And we have like a ground control that we compare them to, and how the plants are dealing in this novel environment. So here is an example of what it looks like. You have plants grown on Earth, and those are our ground controls. And then you also have plants grown in microgravity on the ISS. And you can see that the roots are growing all over the place. That's because there's no gravitational pull to tell them where up and down is. And the interesting thing we learned about such experiments is that in the absence of gravity that tells the plant where is up, where is down, light becomes the cue to tell them how to grow. So plant roots are usually negatively phototropic, so they grow away from the light. But then in spaceflight, they use light as the cue to figure out where up is and where down is, which is pretty interesting. Yeah, so, once we get those plants back from station, we extract the RNA, DNA, and then investigate the genes. How do plants respond? What kind of genes turned on? So with those information, you can understand if they are experiencing hypoxia, if they are experiencing flooding, drought, all the different stresses that that are important for us to grow plants successfully in that environment. So here the bars that you see in red tells you that the genes are turned on. The bars in green tells you that the genes are downregulated, so they are kind of not expressed at a lower than normal level. Yeah, so the interesting thing that we found as well is that leaves and the hypocotyls and the roots, they all respond to the spaceflight environment very differently. You can see all the genes in some examples over here. These set of genes are turned down, these set of genes are turned up, and in the same area you can see some up in leaves and roots and down in hypocotyls. So the overall story that we've been getting with multiple spaceflight studies is that plants are experiencing these the genes that are changed in spaceflight are associated with things like cell wall remodeling. So the cell wall of the plants, those genes are being changed. Reactive oxygen species, these are kind of genes associated with stress signaling. The plants in their environment, in the normal environment, are using genes that kind of simulate a stress response. We have redox signaling, cell polarity, light signaling. Again, because in the absence of gravity, light signaling becomes like a huge cue for them to decide how to grow. So we see a lot of light signaling responses in plants going on in space. The other really interesting thing that we've found out is that from several of our data sets that there are many unknown genes that are changed in the spaceflight condition compared to the ground control. So a lot of unknown hypothetical genes, and this is interesting because this particular aspect was basically what I did for my dissertation. I was really intrigued by the many unknown genes that were induced in the spaceflight environment. Actually, there was something like 50% of one of our data sets were basically unknown genes. We had no idea why so many of them were being used for the plants to adapt to this environment. So for my dissertation, I basically had this obsession of trying to figure out what one of these genes, what the function is of one of these genes, and why the plants are using it in the spaceflight environment. And so basically that's what I did from my PhD. I just tried to figure out this function of this unknown gene And, you know, because you discover it, you get to name it. And I basically named it OMG1. This is OMG for the many OMG moments, good and bad, in the lab. But it turns out that OMG1 is is important in helping the plants regulate their reactive oxygen species pathway in spaceflight. So it kind of is a gene that that the plants use to deal with the stresses in space. Specifically, what kind of stress is still to be determined. Yeah, so you know, the other really cool thing about growing plants and investigating the molecular aspect in the spaceflight environment is it leads to really interesting discoveries that might be masked on Earth. So yeah, this is a list. Well, it's not a complete list. It's just an example of the different type of plants that have been grown on the ISS. There's a whole bunch of them. Also here I show you the different type of hardware on station that have been used to grow these plants. I'll be talking about one in particular, and this is called the Veggie Hardware. So scientists and engineers at Kennedy Space Center have developed this hardware, the vegetable production system hardware called Veggie. And in 2014, Veggie was installed in the Columbus module of the ISS. So here is an up-close image of lettuce and Mizuna plants growing in the Veggie hardware. And astronauts have eaten the lettuce that was grown in Veggie. So, you know, as we embark on our preparation for the moon and Mars, plants are definitely paramount to our space exploration agendas. So the main thing that, that we want to make sure that plants can grow well in this environment is because, of course, food production, life support system, and also psychological benefits for the crew. Many of the crew on station have noted how much they like taking care of plants grown on station. And so right now, I think at Kennedy Space Center, they are actually doing a follow-up with all the astronauts that have handled the plant experiments on the ISS. They're doing a survey to quantify and to really get a good handle of how the plants make the astronauts feel. You know, do they have a therapeutic experience when they harvest the plants, when they deal with any plant experiments? Do they like it, that they have to water them, take care of them? So these are all currently being evaluated at Kennedy Space Center. All right, so then I'm going to shift gears a little bit and tell you about one of the big events that happened on the ISS that led me to become really interested in understanding the plant microbiome. So in 2015, Xenia plants, these are flowering plants, were grown on the ISS and in the Veggie hardware. And these plants were infected by a fungus. You can see over here some of the fungus mycelium that are growing out on the plant. From when they found the symptoms to when it was actually diagnosed took about 12 months. They had to harvest the plants, send it down, and then scientists underground started to diagnose the pathogen. So it took a really long time for the symptom to occur and for the diagnosis to be made. The source of the contamination is still unknown. Nobody knows where the fungus came from. Was it something that was already in the plants, and because the environment altered, it went from being a commensal microbe to a pathogenic microbe? We have no idea. So this fungus actually is it's called Fusarium oxysporum, and it's an opportunistic fungal infection. Basically, what happened, I believe what happened was there was a malfunction in the fans on in one of these veggie units. And because the fans weren't blowing, there was a buildup of water around the plant base, and that environment made it possible for the opportunistic fungus to infect the plants. So this is also an example why it is very important to know the plant microbiome, what the basal plant microbiome is, and how it alters in the microgravity environment. So the one bad thing about Fusarium oxysporum is that it is a pathogenic fungal strain that can cause Fusarium wilt in almost every major agricultural crop. So it's a really bad fungal infection, and it can also infect mammals that are immunocompromised. And that's why you see Scott Kelly over here with a dust mask handling the infected plant samples as a precaution. So yeah, we need, it's not just about understanding the how the microbes affect the plants, it's also protecting the crew, because pathogenic fungus like these can infect immunocompromised astronauts. And we want to make sure that when we do grow plants, we are not putting the astronauts' health at risk as well. So the plant microbiome is, just like the human gut, has a lot of important aspects, as in many of the surface symptoms that you see in plants can be attributed to the microbial activity within the plant microbiome. So things like, yellow leaves, or tolerance to environmental stresses, disease susceptibility, all these are important to, all these can be attributed to the microbial activity. And these symptoms have root causes. So if you have an inadequate microbial community and activity, then that can lead to poor root development, nutrient depletion, and inadequate water uptake. So that's why it's also really important to study these things. But again, it's not that straightforward, because the plant microbe relationship is very dynamic. You have commensal microbes that can become beneficial or can turn pathogenic based on the environmental conditions. So things like the temperature, the pH, how much water they're getting, the agricultural practices, all these things can influence the plant microbe relationship. A lot of studies have been done on Earth, and it's we're still learning a lot. There's a lot we don't know, and we're still learning a lot about how the plant microbe relationship works. On one of the really good examples, or the most well-studied relationship that we have right now about the plant microbe dynamic is in the rhizosphere of the plant. So this is basically the root zone area. In your typical plant growth environment, which is plant growing in soil, there are thousands of microbial species such as bacteria, fungi, protozoa, nematodes, earthworms. These things all play a role in boosting the plant health. So, for example, if you're talking about fungi produces like hyphae, which moves the soil around and that improves the soil structure. Things like bacteria break down organic matter and release nutrients for the plants to take up. And you have other microbes like nematodes, protozoa, and they consume the bacteria and the fungi, and then their waste products act as nutrients for the plants. So it's a cycle of nutrients. And they also, when they're eating the bacteria, it helps with disease control as well. So it's a very dynamic relationship plants have with their microbes. So how do you study the plant microbiome? Studying the plant microbiome requires a lot of sequencing. So what we do typically in the lab is we take the plants, we look at the leaf microbiome, it's called the phyllosphere microbiome, and then we also look at the root microbiome. Again, most of our plants at Kennedy Space Center are grown in arsenite. So we have to remove the clay substrate and then study the microbes that are on the roots. So we take the DNA, and the DNA basically leads to understanding the metagenome, and the metagenome tells you what the microbial community is like. So basically who is there in the community. And then we also do an RNA section of the analysis where what we're doing is looking at the transcripts from microbes. And then that basically leads to metatranscriptome, which tells you what they're doing, what the microbial community is doing. So we do a little bit of both to get a very comprehensive understanding of the plant microbiome and the activity. So on station specifically, it is because this analysis requires so much sequencing. On station, the usual platform for sequencing is called the Illumina platform, and this is a huge machine. So it's not really feasible to bring up to station and use it for the sequencing that needs to be done to do such analysis. So there is a new platform called the mean ion sequencing. And it's basically the future of sequencing in space because of its really small mass and footprint. You can see in this red box the size of this sequencing platform. It's the size of a candy bar. It's really tiny. And so, you know, astronaut Kate Rubins in this picture over here has been doing the experiments with the mean ion on station. However, so far, most of the experiments with the mean ion is looking at the microbes on station. So mostly microbes of the astronauts eating surfaces. So they're tracking the microbes that have gone from the dining table where the astronauts eat to the walls, and they're just tracking to see what kind of microbes are over there. So they've not really used on station exactly the mean ion to do host microbial analysis, not yet anyway. But that's the plan. That's the plan, to have a sequencing platform on station so that host microbial analysis can be done, microbial tracking can be done on station without having to bring all the samples back to Earth, process it, and then figure out what's going on. Yeah, so for my postdoctoral stint, I basically optimized a method to sample the plant microbiome with the MinION. So I use the the MinION, even though it's really small, there's a lot of steps that need to be done so that you can effectively figure out what the plant microbiome is like. So what I've basically done is developed a protocol that will let you study the plant microbiome efficiently using the MinION. So this is something that I'm hoping that we could implement on the ISS. So here's an example of some of the data that I've obtained. Basically, using the MinION sequencing platform, I can figure out all the different microbes that are on. So in this particular experiment, it was looking at the lettuce, what's on the leaf surface. We decided to use the lettuce because it was the crop that the astronauts ate. And we wanted to see on the surface of the lettuce, what kind of microbes are there and what they're doing. So with this particular methodology, I was able to find out what microbes are on the lettuce. And we found potential plant growth promoting bacteria. And so these are a few of them highlighted in blue. And then we also found potential pathogenic fungal genres in our data set. And these, again, we mentioned Fusarium. Fusarium was also present in our data. So we want to know whether, the thing of it all is with bacteria, just looking at the genus, you can't tell if they are pathogenic or not. And it depends on the strain, and sometimes it also depends on the environment. So, like, for instance, the opportunistic Fusarium oxysporum, it's not always pathogenic, So just based on this sequencing data, they have the potential to be. So these are things that we are paying attention to. What genuses, what genres are in our community, and how we can monitor their development as the plants are grown on station. So, with this pipeline, the other thing that you can do is also find out what the microbes are, what the microbes are doing. So with this pipeline, I was able to find with the bacteria community 106 different pathways that are active in the community, and with the fungal community about 85 pathways. So you can see that the microbes these are some of the key functional activities that we picked up. And what we can see is that the microbes are engaged in environmental adaptation. So we see cell growth, cell death, signal transduction. These are things where they are taking cues from the environment and they are reacting to it, and many different other metabolic activity. So we can see in this particular instance, a huge number of pathways are associated with the signal transduction. So that means that the signal transduction systems enables bacteria to sense, respond, and adapt to a wide range of environments, stresses, and growth conditions. So they are noticing that they are in this different environment. The other thing we can do is then look at the individual genes involved in the specific pathway associated with signal transduction. So such information can tell us what genes we need to pay attention to. The behavior of those genes over time can then kind of guide us to identify important drivers of change in the microbiome. So for instance, here, you can see a tight cluster of genes that are associated with the MAPK signaling. So one of the really interesting things about the MAPK signaling is that it's involved in fungal pathogenesis. So monitoring the plant microbial function can also help us detect signals of distress in the plants. So in the data that we have, we can see some, like I mentioned, some potential plant pathogens. So then the question becomes, what kind of approaches can we use to combat potentially pathogenic microbes? So there are, more and more studies are showing that you can use beneficial plant growth promoting bacteria to combat these microbes. They found that in certain bacteria such as Bacillus over here, Bacillus actually produces a compound that can eradicate fungal pathogens like Fusarium. So we basically need to really understand what is present in our plant microbiome so that we can figure out how to use them to help ensure successful plant cultivation in station or on Mars. So like I said, the whole process is not as straightforward because there's still so much that we don't know about the plant microbe interaction. But however, there is a big push in the scientific community to understand the interaction between plants and their microbes. And here are some of the examples. More and more papers are actually being published on how certain beneficial microbes can help promote plant growth and also inhibit plant pathogens. So eventually, the hope is to be able to understand it enough so that we can exploit beneficial microbes for our spaceflight application. So what's next? Well, one of the main things that I'm working on is to automate this methodology that I've developed with more microfluidics, and so that this process can be easily performed on station by the astronauts. And I think it's really important because, you're going to be traveling to Mars, and it takes about 9 months to get there. And in that time, you want to know how well your plants are growing. So what happens when the plants grown are infected by a pathogen? How do we determine what's causing the infection? There's no way to send the samples back to Earth. So we need to develop tools to try and investigate these things on site. And then once we find out what the microbes are and what the causative pathogen is, how do we mitigate these problems, so that we minimize the risk of losing crops and ensuring the crew members are safe. So these are really important questions. And the other thing we want to do is also start using spectral imaging to monitor plants on station, and also in concert with monitoring the plant microbiome. So things like hyperspectral imaging or multispectral imaging are things that we are playing around with right now to try and see how that can give us some insight into what the microbiome is like as well. So if you see a plant with a lesion on top, we're using hyperspectral or multispectral imaging, can we then use that, can we then pinpoint that particular lesion, do the microbiome analysis, and find out what's causing that lesion? The other thing is that we have talked about also is using AI to kind of monitor the changes in the microbiome of these crops over time. And then could we then use AI to help us develop predictive tools on how to better manage the plant microbiome so that we prevent the formation of plant pathogenesis. Yeah, and also using the same thing with AI to predict what kind of microbes will help mitigate these potential problems that we see in crops grown on station. So yeah, so that's more or less my presentation.
Thank you, Natasha. That was a wonderful talk with a lot of insights on the microbe in the space. Good to know that you have discovered a new gene, called it OMG, and you have a sense of humor too. And it was nice to hear some of the
yeah, as a scientist,
places.
I think you have to have a sense of humor. So I should be kind of depressed.
Yeah. So I was wondering about the COVID situation. Is there any impact? Is there any stress is shown to COVID kind of virus? Does it affect plants also? I mean, I'm just curious.
Yeah, there are quite a few viruses that can cause lesions in plants and stuff. So one of the things I'm actually in the process of writing a proposal for is to test out this MinION system on viral plant pathogens. We have fungal, and we're trying to do bacterial. So we want to have a whole slew of different pathogens that we can just use this one system to detect. Yeah, so we'll see how that goes. But yeah, that's kind of, we definitely want to make sure that we tackle viruses as well, because a lot of plant pathogens are viruses as well.
Oh, interesting. I will open the floor to question answer session. You can unmute. I can unmute everybody, so you can start asking questions.
Okay, can I go first? Natasha, this is Venkatesh. It was a wonderful presentation, very exciting to hear some of the things that's happened and in the last few years. I had two unrelated questions to picking on a couple of things that you said. One was you talked about plants providing psychological benefits for the crew.
Yeah.
Are those is that study or analysis just based on? Is it just being based on just a survey of the astronauts? Is that fundamentally how they're doing it, or is there more rigorous studies that's going on beyond that?
Yeah. So for now, Yeah, so for now, most of the insights that we're getting are tweets that the astronauts send out that, oh, I'm so super excited to, to eat these lettuce or to harvest this lettuce. So a lot of that is like from social media platforms and from them talking to scientists at Kennedy Space Center saying, oh yeah, that was fun, I really liked it. But right now there is going to be a huge effort to try and really quantify that. So astronauts that work on plant experiments on station have to fill out survey forms. There is taste tests that is being done. There is like if they're handling the plants, how do you feel about cutting the lettuce? How do you feel about watering the plants every 2 days? How do you, a lot of questions that are being asked to the astronauts right now to really get a good quantifying matrix for how it really benefits them.
Because there are other studies that actually NASA has conducted on the benefits of plants that I think are based more in Earth, if you will, and those studies are bandied about quite a bit, and very interesting analysis from that. So so that's why I was curious about whether they're using similar methodologies out there.
Yeah.
Okay.
I think that's a big push to that.
And then the second question I had for you was much more broader. Are there lessons that that are being taken from these exercises and experiments that's happening for space, and trying to bring it back to Earth plants? Is there reverse going on?
Yeah. So, for example, this pipeline that we've developed. Even though the idea was for spaceflight application so that we can test figure out what the plant microbiome is like, and to automate that. The idea is if we can come up with a shoebox hardware that does it all, then you could definitely take it to the USDA ports where they screen for pathogens that are imported to every different country kind of thing, right? So right now what they do in the field to determine whether you have a pathogen or not is through this ELISA kind of paper thing. So if the pathogen is there, then it will send the sign and say that, oh yeah, this strip of paper will show you, okay, yeah, this pathogen is present kind of thing. But it doesn't help you discover novel pathogens, something that you don't have a developed kit to test for. So things like that we can definitely use. Even though the application that it was developed for is for spaceflight, we can bring it down and use it on Earth for sure.
Thank you.
Yeah.
Hey, Natasha, I just had a question as well. Just in terms of how you were able to conduct your research using the Martian stimulants, what were some of the considerations you had for the type of soil that you used as a stimulant? And the second question that I had was regarding whether plants were able to sort of live without any bacteria. You mentioned that there were some bacteria that was helpful in terms of being able to sustain the microbiome, like having plants without that. So those two-part question, if you can.
So the first part, a lot of that research was done at the University of Florida with the PIs, Dr. Rob Frohl and Dr. Annalisa Paul. And over there they had, I think what was available to them was those two Martian stimulants, the one from from Hawaii and the other one from Mojave. So I think in that lab, the experiments that we did with those two stimulants was because that was what was available for us to grow the plants on. And then to your second question, I don't know if plants can survive without microbes. I mean, it's just like the human gut. You have microbes lined in all over. You know, the plants have the same thing too. It's on their leaves, it's on their stems, it's in their roots. It's all over. There's also a airborne microbial, they call it. So you have the plants growing in the soil. You can actually sample the air microbes around the plant. So it's all over. I don't think they could survive without it.
Oh, okay. So there's yeah, absolutely. So does that mean that you can't really have a full vacuum to experiment whether plants will grow in it without any of the bacterias available?
I don't know. I don't know. I don't think I've seen personally. I don't think I've seen a paper on that kind of experiment. But you know, I don't really know.
That's okay. That's
Can
cool.
I
Thank you
jump-
so much for the talk. Very cool. Thank you.
Yeah.
Can I jump in and answer her question?
Oh, there you go. Yes, please do.
So there is a paper out there. You can pick it up on ResearchGate, and I will actually drop the link into the chat on using the Mojave simulant specifically for the Mars analog, since that's the one that you used at U of F. And you always have some kind of a biome. It just depends on what quantities of each particular aspect you have available to you, because the plant has its own, the water has its own, the soil simulant that you might use has its own. And just like how you would have a natural yeast in your air, you're going to have whatever oxygen supply that's being utilized will have its own biome as well. It's probably easiest to think about a biome as the house, and whatever is being grown inside it will depend on where it's being located. So you always have something. What we have to do as scientists is kind of figure out what's there and how it's going to affect it. But this paper is really useful, and hopefully it'll help you guys out.
Nice.
Thank you so much.
Thank you.
Natasha?
Yeah.
I just had a question. Have there been any experiments in growing a really small tree in space?
A small tree in space. Well, actually, what they have done is experimented with peach trees. So peach is something that it's definitely beneficial for the astronauts because it helps with their bone, the bone density loss. So eating peaches are definitely helpful. And so what they have actually done is an experiment where they keep touching the tree. So touch response is something that plants definitely react to. And so what they found is actually if you keep kind of giving the plants this touch response, instead of going upwards like a tree, it kind of grows, it kind of sprawls outwards. And so it's something that they've been thinking about, you know, using that kind of method to kind of grow peaches in your very enclosed environment. Because it's definitely beneficial for the astronauts to have peach in their diet. However, I don't think they've done those experiments. I think on Earth. I don't think they've actually done it on the ISS yet. Yeah, but there is definitely that that kind of experiments that are ongoing with trees.
Maybe bonsai, I forget exactly how it's pronounced, the miniature trees.
Oh, the bonsai trees?
Yeah.
Yeah, I don't know. I mean, you definitely want it to be of a decent size so that it can have decent yields of fruit. The really interesting thing about the word is kind of slipping my mind right now. The touch response basically is that because it's kind of sprawling outwards, it also produces fruit continuously. It's some kind of it is really interesting. The USDA actually was did a study with one of the NASA scientists, and they're trying to figure out how they can implement this in station. So fruit, it doesn't become a tree, it kind of sprouts out like a bush, and in a confined environment, this is perfect. And then on top of everything else, it also produces fruit continuously. So it's really interesting.
That's fascinating. Could you point a reference to this touch response research or a survey or something?
Definitely. Let me get.
Thank you. Thank you very much.
Yeah. Mm-hmm. Yeah. Well, I find that paper, if you guys want to keep asking questions about me.
All right, thank you. This is totally fascinating and new to me.
Awesome.
Could I ask more of a personal question, Natasha, in terms of like how you got into it and what drew you into the topic of creating or being able to grow plants in space?
Well, so I'm initially from Singapore, and then I had an opportunity to do an internship over in Boston. And, since I was 9, I always wanted to be an astronaut. But Singapore doesn't have a space program. So what I had to do was, change my childhood ambition from being an astronaut to a scientist. And then when I came over here in the US, I had the opportunity to meet with Rob and Annalisa and realized that they were all about growing plants in space, and that captivated me. We had a really great interview and had really great conversation, and I knew that was what I wanted to do. I mean, my interest in space and being an astronaut kind of just made sense. So yeah, that's basically how I got into this, and been really happy in this field.
I've got a question for you. Steve Adams from the University of Oklahoma. Are you aware of anyone who is growing or doing experiments with centrifuges to induce some gravitational effects on plants?
Oh yeah, definitely. There is a lot of clinal rotation experiments, 3D random positioning experiments that are ongoing right now at Kennedy Space Center. So some of the simulations that gravity simulations that we had to do were also trying to grow plants in a clinostat. Centrifuges as well. I know John Kiss's group out in where is it? I think it's out in Greensboro in North Carolina. He basically did a plant experiment where he grew plants in this hardware that was a centrifuge, and he did comparisons of that experiment in station and in this centrifuge device. So you could basically see simultaneously how plants respond to the lack of microgravity and the artificial gravity.
Thanks.
Yeah. How do I, nope, not that. How do I stop share? There we go. All right, perfect. Yeah, let me just, I have that paper somewhere in my hard drive. I need to hook it up.
So one quick question, Natasha. So you have mentioned peach is one of the favorite food there. Is there any top 10 food for astronomers they use? I mean, favorite food.
Well, lettuce, definitely salad crops for now, because salad crops do not require a lot of processing for consumption. You pluck and you eat kind of thing is basically what it is. I mean, they do a little bit of sterilization, but for the most part, you don't have to cook it or process it in any way. So right now, salad crops are one of the more favorite crops that they want to grow on station. However, they are actually doing a huge survey with the Charlottesville Botanical Center down in Miami, where they are testing and they're having kids from all over the country figure out what what crops grow best in different conditions, and that kind of tells them, if they find a crop that they've basically left in the windowsill for days without watering, and it does a really good job, then they're kind of gonna look at that crop and say, okay, maybe we can see how this will, be used in a flight mission in case of, drought or something like that. So there are a lot of active, citizen science programs, outreach programs that are trying to kind of pick out the best crops. Also, nutritional assessments of crops are being conducted. They are trying to do a good survey of what nutrients are available in what crops, what nutrients are necessary in the astronauts' diets, how we can, cross-compare which ones provide the most nutritional value. So a lot of that kind of research are actively going on right now.
Thank you.
Yeah.
Can you guys hear me?
Yes.
Okay, cool, awesome. So kind of related to that last question you answered, I wanted to know how you see CRISPR being involved in the near future of engineering the most optimal plants that we can send to Mars. Like, even though we don't know the whole genome of anything, if we can figure out how to insert the genes from like that window resilient plant that you were mentioning, or like the genes from Chernobyl fungus that thrive with radiation, and then insert those genes into some edible plants so that it becomes more resilient against the space conditions. Like, where do you feel our knowledge is about all that stuff needed to do all that?
Oh, there's still so much to discover and to really understand before we can get there. I mean, CRISPR is definitely going to be used to genetically engineer plants in the most precise manner. It's just that, you know, in terms of what we know about plants, the molecular response of plants in this environment, we see big pathways that are being altered in spaceflight. However, you know, every time you look at the genes, the specific genes in that pathway that are affected by spaceflight, it's not always the same. It's very dynamic. You know, the plants are amazing because they have so many redundancies in their gene pool. They have so many genes that do the same thing, and it's because, you know, they can't move. They are sessile. They have to quickly respond. Even like, you know, the car light that is being flashed when a car drives by and your headlights are flashed on a plant, they respond to that, you know, in very complex manners that to try and say, hey, this one gene is the gene that we need is a bit difficult. I don't think it's that easy. So there is still a lot that we need to learn. I mean, there are, you know, genes out there that, hey, you know, if you modify this, you get this outcome, this phenotype, and this phenotype is beneficial for applications in space. Those kind of things are definitely ongoing, and that's what we're doing. We're trying to look at, you know, we collaborate with USDA to figure out, you know, what they know about plant diseases, what they know about, you know, genetic changes in plants that, you know, yield continuous food production, smaller mass, biomass, less waste, so that, you know, if you grow a crop, it's not only growing the crops for consumption and for life support, you have to figure out what you're going to do with the waste of the crop when you don't eat all parts of the plant, right? So those things are stuff that the USDA and NASA are working actively to kind of resolve. But there's still so much we don't know, and there's still so much to know. So yeah, it's a good question, but I don't know if I have a really good answer for you.
Well, so basically we don't know a lot about the genome, and we don't know a lot about how genes, like what genes will do in plants, because plants are just so awesomely complicated.
Yes, exactly. And then plants aren't like mammals, where we only have 2 copies of our genome. They have sometimes all the way up to 8, 16. So they are really complex.
Yeah, I was visiting this Hawaii. There was a tree which grows in volcano soil, and it has a different way of adapting to this volcano rocks. So what it does, it grows small tree above in the top of the tree itself. So it drops the small grown plant on the when there is a good soil, it drops them. Otherwise, if it is not conducive, it keeps that small plant in itself. So it is already grown plant. It grows on the top of the tree. Whenever there is a good soil, it drops that. So I'm not sure. Is it that way? Martian soil, we can use the trees which is grown in volcano region, like in Hawaii, they have this type of trees.
Yeah, I know there's a lot of different possibilities. Right now, from just, you know, in just talking to people and stuff, I think the emphasis is to kind of work with the crops, engineer them if possible. Still a lot that we don't know, but if we can figure out, I don't know if we're gonna be, if I understood your question correctly, I don't know if we're going to be able to, you know, manipulate the surroundings so much. I mean, with the soil and stuff, we could definitely add microbes, add nutrients and stuff like that. And I do believe a lot of people are working on that kind of experiments and trying to get, you know, data about, you know, what needs to be supplemented, what needs to be removed, even microbes as well. Like, you know, they're trying to figure out how best to deliver a consortium of microbes, because microbes, the other thing about microbes also is that one organism by itself, when you deliver it to the soil to supplement, doesn't always work out the way you think it would. It needs like a community to thrive sometimes. So it is really complicated. I mean, you could definitely do those kind of analysis, and a lot of people are working on it to try and see how you can change the how do you not change, but rather like supplement and improve the conditions for plants to grow. I do believe there was someone who worked, what's her name? Tamisa? I think she was working with the Martian regolith, right?
Yes.
Yes. Okay. Yeah. Did you have thoughts about that? More?
I'm sorry, can you repeat the question?
Did you have more thoughts about what kind of supplements are good for trying to grow plants on those simulants?
Yeah, so it depends on which simulant you're using. If you use the Mojave simulant, they've got two different levels, MMS1 and MMS2. MMS2 is the closest analog we have to Mars, but you still have to tweak it a little bit by adding specific chemicals, perchlorates and so forth. Once that's achieved, then you can look at which plants work. So I'm actually doing potatoes, so I'm playing Mark Watney.
Nice.
And yeah, so it's kind of fun. Each plant has a specific nutrient requirement, and you have to keep that in mind when you're working at it. So you can't just use like a generic garden, you know, 7:10 NPK style ratio. You have to look at which plants you're growing, what type of water requirements, what type of light requirements, and what type of nutrients. Once you've figured that out, and we have a lot of that data because we've been farming for many, many years, just in general, you can go in and create what we call a plant library. So corn requires certain things, potatoes require certain things, and then it's just putting the right chemicals into the water and making sure that your water ratio and your light ratio and your nutrient ratio works
out.
The problem with the lab is, of course, we're on Earth, we're not on Mars, we're not on the moon, and therefore we can only do so much. And then we have to try and convince somebody at NASA to send it up to space and see if it still works. But if you were to do like lettuce is what we use a lot because it's got a very quick turnaround. It's like radishes. It's a very easy plant to grow.
Right.
You have certain nitrogen ratios and certain potassium ratios. You have a little bit of iron and magnesium that you need. Most of the minor micronutrients is what you can find in your soil, but in a place like Mars, those numbers are not the same. So whatever we're growing has to match either what the soil has or the regolith has, or you have to add it in. So that's whenever you get to all these fancy gels and liquid nutrient fertilizers that have to be added into it. But it's all dependent on your plant that you're trying to grow. I don't know if I rambled. I'm sorry. Hopefully that answered the question.
No, that's great. Yeah. No, it is very, it is not as straightforward because, you know, every plant has very different needs. You know, temperature, pH, all these things affect the plant growth as well. And so, yeah, I can only imagine how huge your database must be with all the different things that you probably have to supplement or, you know, for each different plant.
You can try and do it yourself at home. You can buy the soil simulant and a bunch of LEDs and try it yourself. It's a great citizen science project. Yeah. And if anyone's ever interested, you could just type it into Google and you'll get all sorts of stuff. But it is a very fascinating field, and you can make it as complicated or as simple as you want because somebody else will always come and complicate it after you.
Of course. Nice.
I'm curious to know what kind of salad greens are considered the best for, you know, people on space flight. Is it like kale that everyone here thinks is so great, or are there something else that's like more nutritious or is considered better?
Oh, that's a good question. Unfortunately, I do not have the answer for you. I do not know which lettuce, what the nutrient you know, content for each different lettuce is. Yeah, no, I do not know. But I'm sure there are people out there that, you know, are looking into the nutrient content for each of them. I do believe that actually this is something that NASA scientists out at KSC they have like 4 or 5 different lettuce cultivars that they're trying to get a good or a holistic nutrient profile. But yeah, I don't know the answer to that.
I've got a question about algae, like spirulina. So like things like algae and spirulina, I hope I'm pronouncing that right. Spirulina, do you know what I'm talking about?
Mm-hmm.
So
Yeah.
Spirulina, it's like a type of cyanobacteria or algae or something. But what's good about it is that you can actually eat it. Like my mom buys the powder of it, and it's a great source of protein. But what's good about it is that it doesn't take a lot of water to produce it. So is there any like technically, well, I don't know technically, but algae are like some sort of plant. So can you grow that in space and for Mars as a means of getting your protein?
There are a couple of algae experiments that are going that are ongoing right now. A lot of them are being used in terms of biofuel. I know one professor at the University of Florida actually just got awarded a grant from NASA to kind of investigate how he could grow this algae to produce biofuels. In specifically for spirulina, I do not know, but yeah, that'll be interesting, you know, prospect to investigate for sure. Mm-hmm.
Do you know what that substance in peaches is that's supposed to help reduce bone loss?
Not at the top of my head. I did send that link to that paper. I have to read it up myself. I can't remember what compound specifically is helps with bone loss. But yeah, yeah. Do check that paper out and see.
I mean, wouldn't it be easier to just give them some supplements with whatever that thing is than to try to grow a peach tree just to get that one thing?
Well, yes and no, I guess. I mean, it's also more of a can we use plants to kind of not only just create a sustainable environment, right? But can we use these plants to supplement nutrients for the astronauts? And if we can, why wouldn't we try and, have these plants do most of the work where you're creating a sustainable environment, you're producing food, and I think this, although the peach trees are a really interesting prospect, I don't think it's something that we're going to do in station. It's kind of more in terms of when we do go and onto a planet and we colonize it, here are a few things that we can grow on the planet, kind of thing. In station, I think it's hard for them to grow a peach tree, even though it's more sprawled out and much smaller in height. Yeah, I have let me see. I don't know if I can share this. Let me go to the screen and show you what it looks like. Oh, I don't think I can. Oh yeah, I can share my screen. So here, for example, is a picture of the peach tree that yeah, okay, here. That this is, oh, sorry, not peach, I mean plum. Got them all mixed up, sorry. It's Friday. This is the plum tree that usually grows to be about 2 meters in length. And with this gene, you can get it to sprout out like that. And it also produces continuous fruit. So yeah, that's why it looks like from a huge tree to a little kind of viney bush. It's pretty exciting. Yeah.
So anybody else? So one quick question about I mean, synthetic protein, like here they create synthetic meat and something like that. Are they, how they get their protein? I mean, fruits, okay, vegetables. So how they get their protein? Is there any algae or anything they use to synthetically create protein?
I don't think I understood your question exactly.
Yeah. So far I heard about fruits and vegetables, and what about the protein the astronomers has to get? Is there any plant-based protein they're getting, or?
Oh, in their diet right now?
Yeah.
So most of their food packet are kind of pre-packaged, so they are like rations that they have. So they do eat. They have like meat and everything that they have in their rations right now. Plant-based specific. I don't really know exactly what is in their food packages. I believe there should be quite a bit of vegetables and stuff like that, but plant-specific protein, I don't know. Don't know enough to say what exactly they have in their prepackaged meals that they bring it to the station. But the idea is that if we can grow fresh fruits, then fresh food for the astronauts in station, then yeah, it'd be great if they could get some kind of plant-based protein for sure.
Okay, thank you.
Do you think it's practical to engineer a plant using maybe CRISPR or something else so that it doesn't need a microbiome? It's sort of whatever the bacteria was doing to help it, you just kind of put that in the plant and then you don't really need the bacteria. Is that even remotely practical, do you think, or do you just really need a whole community of different bacteria?
Bacteria is everywhere. And the interactions with that the plants have is, it definitely is. It's going to be there. There's so much. I think your question is if we can just get the important aspect of the bacteria and just give it to the plants. I don't know. I don't think that biologically that's going to be feasible, because everything has some kind of bacterial population. If the bacteria helps the plants, we should definitely investigate that and see how we can monitor it. I don't think getting rid of the bacteria and just using the compounds it produces and supplemented the plants is the way to go. Yeah, that's my opinion.
So what I was thinking was just to kind of simplify the process of growing something. If you have to have a whole community of microbes, it seems like a much more complicated thing to make work. Whereas if you could figure out, well, this bacteria is helping in this way. I mean, the reason the plants have the microbiome is just because they're growing in bacteria, bacteria is all around them where they're growing. So they just evolved to get used to that. But in space, you could create whatever condition you want. It just seems like it might be simpler to not need 1,000 different microbes to keep the plant happy. If you could make a plant that only needed, a couple microbes, it just seems like it'd be a much simpler thing to get working.
Yeah, I see your point. But you know, it's like it's like your gut microbiome, right? There is such a huge community, and every single microbe does something. It's really hard to be able to figure out what each microbe does and supplement those compounds to your gut lining. In that train of thought, it is the biology of how things work. Even in the seeds itself, they have a microbiome. And people are studying the microbiome of individual seeds. So it's not like it's something that, you know, is because it's everywhere, they get used to it. It is kind of in their evolutionary development to have that microbiome, just like us, like with our gut lining. It'd be hard to just take all the microbes out of your gut and find a way to kind of supplement their compounds that they produce. You know what I mean? That's my thought on it.
Yeah, I mean, especially for the gut, I could certainly understand that would be really, really hard to do. But I guess I just didn't really know much about plants. I just kind of was assuming maybe they were simpler than the human gut.
Yeah, it's a very complex system that they have as well. Very simple. It is the microbiome that plants have. It is pretty complex, and it's still a lot that we don't really know about. Yeah.
Thank you, Natasha. I think it is almost 2 hours. It started. It is going to be 10. It is late for you also. It is Eastern time. It is 10, I think. I'm also in Eastern time, Boston, close to. So we can conclude that. If any questions, anything, anybody last chance to ask one more question. Okay, I think everybody's satisfied, and we can conclude this session. Dave, you want to say some words?
Yeah, I'll just really quickly. I want to piggyback up what the other Dave was just saying. That was an awesome conversation. I had to step away for just a moment, but I'm really glad I picked the end of that up. And I don't think that that's totally off base. I think that, you know, maybe if it's not that you have a couple of microbes that are, you know, are a plant that only needs several, but maybe you can kind of, if we're engineering plants and we're engineering human, maybe we can engineer microbes so that we can kind of come up with a catch-all that provides the nutrients, you know, kind of picking and choosing, you know, carefully to make sure that we can take care of each critical plant's needs, you know, and craft the microbiome from the ground up like that. Anyway, but so just want to get that in there. Dr. Natasha, thank you so much for this wonderful presentation. I have probably a million questions for you myself. We'll have to pick this up another time when it's not 10 o'clock on a Friday. And and as always, Zen for Quantum is providing the best content. Love it. This is so interesting. Thank you, Balaji, so much for putting this together and having us all here tonight. And
thank you for the opportunity.
I have recordings. I will publish that in the meetup. If any reference you need, you can pick up from there. Thank you. Thanks, Natasha, for a wonderful talk.
Thank you, Natasha. Thank you, guys.
Have a good one.
Yeah, bye.
Have a great day. Bye.
Bye.
Thank you. Bye.
I can only get one link at a time. Is it possible for somebody to save some of them and post them to Meetup or something?